Hello, welcome to BioFree again. Today we'll talk about the eukaryotic cell, specifically the structure and function of eukaryotic cell. We'll focus on the nucleus today. So we have two types of eukaryotic cell, animal cell and plant cell. So this is animal cell, as you should know, and this is plant cell. And both animal cells and plant cells have a nucleus. We call it eukaryotic. Eu means true. Karyo is it means nucleus. So both animal cells and plant cells, they have true nucleus. So that's what we are focusing on today. We will focus on the nucleus of the cell. So the nucleus, this is a very important point. We can find nucleus in all eukaryotic cells. Well, there's an asterisk at the end with an exception. Well, like most of the biology, we have exception in almost everything in biology. Almost, okay, except the central dogma. Okay. So in red blood cell, there's no nucleus. That's why the lifespan of red blood cell is very short. It's approximately 120 days. And the function, okay, it stores and protects genetic material, we call it DNA. We talked about DNA earlier when we talked about nucleic acid. DNA stands for deoxyribonucleic acid. DNA disassembled during cell division. Disassembled during cell division. This is a little bit. Well, this statement is a little bit generalized. I wouldn't say it's a little bit general. I would say condense. DNA condense. DNA condense during cell division to become something that we heard a lot all the time. It's called chromosome. So I would say that DNA condensed during cell division to become chromosome. Structure of DNA. It has something called nuclear envelope, or we can call it nuclear membrane. Nuclear membrane is also made up of phospholipid bilayer. But nuclear membrane is double, so it has a double layer, meaning that it has two phospholipid bilayer. If you see in here, you have one phospholipid bilayer here, and you have a second phospholipid bilayer here. So one phospholipid bilayer here, and then a second phospholipid bilayer here. So the nuclear membrane or nuclear envelope is a double membrane. It has two phospholipid bilayer. Oh, okay. Next slide said it has two phospholipid bilayer. Outer membrane continuous with ER ER stands for endoplasmic reticulum. Specifically, we said that it should be a rough ER. It should be a rough ER. Rough endoplasmic reticulum. So rough ER, okay. Perinucleus space also continuous. Basically, all of them are continuous with the rough ER. Well, you can just assume that okay, the entire nuclear membrane continues with the rough ER. When we talk about rough ER, I'll tell you why. There's a very important principle called structure follows function. Structure follows function. There's a reason why rough ER is a continuation of your nuclear membrane. Regarding to the perinuclear membrane, that perinuclear space, it is a space in between the outer membrane and inner membrane. And we also have something called nuclear pore. Nuclear pore, you do not need to memorize the number, okay, as usual. You do not need to memorize the number. The pore connects the outer and inner membrane, yes, but I would say that the pore allow messenger RNA, yes, I think that's a good way to put it. The nuclear pores allow the messenger RNA to go from inside the nucleus to the cytoplasm. Remember the central dogma, we talk about DNA transcribed into a messenger RNA and messenger RNA translate into polypeptide. This is the messenger RNA I'm talking about. DNA is located inside the nucleus, so after the transcription and the messenger RNA is made, then the messenger RNA will go from inside the nucleus to going through the nuclear pore to go outside of the nucleus. And outside of the nucleus is cytoplasm. And then we have something called nuclear lamina. Nuclear lamina, they are the fibrous layer under the inner membrane. That means they are the layer that is in contact with the DNA or chromosome during the cell division. So the picture shows you very clearly, okay, nuclear envelope connects to the nuclear envelope, or aka nuclear membrane, connects to the rough ER. Well, usually people just put an R in front, a small letter R, RER, and then ribosome on the outer membrane. Ribosome, because ribosome is on the rough ER, it makes the rough ER looks rough. So we call it rough ER, rough endoplasmic reticulum, because it has ribosome on the surface. And then when we look at it under microscope, it looks rough. And then we have something called a smooth ER. As the name suggested, the surface looks smooth. Why? Because on the surface of smooth ER, we do not find ribosome. That's why they look smooth under the microscope. Ribosome on the outer membrane. You also have ribosome on the nuclear envelope as well. And then we'll talk about the function of ribosome. It's for protein synthesis, if you remember. Nuclear pore connects to two membranes. Yes, it also connects to the inside of the nucleus to the outside of the nucleus. As you can see here, this part is the you can see that it is the continuation of the nuclear envelope. The rough V out is the continuation of the nuclear envelope. Okay, nuclear pore. Inside the nucleus, we have DNA polymerase for DNA replication. Well, they call it duplication, but most of the texts call it DNA replication. And what does it mean, DNA polymerase? Okay, ASE, as I mentioned before, it means an enzyme. What is the function of this enzyme? Well, look at the name. Usually the name of the enzyme tells you the function of this enzyme. It makes DNA into polymer. So DNA polymerase is an enzyme that makes DNA into polymer. That's why we use DNA polymerase. We use this enzyme during DNA replication. DNA replication is basically making a Xerox copy of a DNA, identical copy. And then we have a RNA polymerase. RNA polymerase is during transcription. We briefly talked about transcription and translation before. And again, the name tells you that ASE is an enzyme. What is the function? It makes RNA polymer. The RNA polymer that we make during transcription, what is it called? Do you remember? Let me give you 5 seconds to think about this. Okay, time's up. The RNA polymer made by the RNA polymerase during transcription, it is called messenger RNA. Messenger RNA or mRNA. Some people just call it mRNA. It should be called messenger RNA. The histone. What is histone? Histone is some kind of protein or histone protein. And what happened is that DNA, most of the time when DNA is found in the cell, they are in the form of chromatin. What is chromatin? Chromatin is basically DNA strand, a polymer of DNA wrapped around histone protein. When DNA wrapped around histone, you may call that chromatin. And then we have some other gene regulating protein that we will not go into details. If you take upper division molecular biology, then it will tell you some example of this gene regulating protein. For example, primers. Primers is one of those gene regulating protein. Outside of the nucleus, what do we have? We have RNA, yes. What type of RNA we have? We have something called tRNA. We have rRNA. tRNA, T stands for transfer. Transfer RNA. RNA, R stands for ribosomal RNA, which is this one here, ribosomal. So you have ribosomal RNA and transfer RNA. Of course, you also have the messenger RNA, because the messenger RNA moves from inside the nucleus to outside of the nucleus into the cytoplasm.